Okay, welcome to the webinar. My name is Klaus Gustafsson. I'll walk you through what we do with ProteinGPS and Infologs and how we use that to interrogate and validate biological systems and navigate in that space. So first I'm going to steal a slide from last week's talk by Brett Lovejoy, my colleague, and really how we see the differences between, on one hand, libraries and approaching sequences, interrogating space through libraries versus interrogating space through ProteinGPS, and it really drives down the difference in the assay capacity and the cost of assay. So the more efficient your assay setup is, the more relevant it is to work through libraries versus if you have a low throughput capacity assay or a very costly assay, you're much bigger advantage to go down through the ProteinGPS path. Another way to look at that is really on this slide we're talking about the cost of assay and as a consequence the throughput of assay for something that's really high throughput. If you want to screen for, say, an improved beta-lactamase, it's very simple to just do simple error-prone PCR, clear it out, and cost of assay is basically zero. And I'm sure you'll find things that grow better on high amp, or if you do try to improve a GFP or something. And just simply cost of generating the library at that point is basically zero. Error-prone PCR is very simple to do. On the other extreme of the axis, you have the ProteinGPS. If the cost of the assay is very expensive, your throughput is very low, you want to make sure that you get as much information as possible out of as few data points as possible, which is the whole take-home message using ProteinGPS. So in between here, I put in a couple of examples, but certainly by no means a full set of alternatives. But really, if you have a medium to high throughput assay, you're much better off doing some sort of antibody-style library. We have certain degeneracy in certain positions, and you screen through that. You either one round or subsequent rounds. Or you can do, if you have a lower throughput, do single-site mutagenesis followed by combinatorial off of your hits from that. And of course, there's a lot of in-between variants how you to approach that, the different ways of doing it. So what I will focus on is the ProteinGPS for the rest of this talk. So what we do here is really using multidimensional regression, and I'll do another 2 slides or so about multidimensional regression for those who didn't pay attention in high school math. So really it's a well-established mature tool. It talks a lot. It really is used a lot in finance and marketing and fermentation and a lot of different data-intensive technologies today. We're using that. And it's within the biotech field, we certainly use it within everything from some DNA array people and QSAR, and lately there's been a lot of work done in multidimensional regressions, multidimensional analysis in the world of SNP and pharmacogenomics and that sort of things. The tools are very nice and mature. They've been around for forever and can deal with a lot of complicated and special issues with large data sets. A lot of the correlations that typically exist in large data sets and how to weed that out and deal with missing data, incorrect data, and so on. And of course, like everything else, there are limitations, but I think here the limitations are basically the same as everything else you do. Of course, you need data, and the more data you have, the better off you're going to be. And always garbage in, garbage out, that's sort of a generic rule for everything. So just as simple, for those who do not know the design of experiment, it really is a question of how to interrogate multidimensional space. And a very simple way of looking at that, if you have an enzyme on the left-hand side, you want to optimize a protein for some given function here, given in the z-axis. And you want to check for, say, 2 different positions, amino acid 1 and 2. And you can really approach it 2 ways. You can take position 2 and make all 20 options, and you get some measurement for some activity, and you pick the best guy, which in this case is this one, and you say, okay, I want to fix amino acid position 2 to whatever this is, and I'm going to do the same for amino acid number 1, and I get a new distribution. So you end up with some data point that has some activity. And of course, you end up doing 2 times 20, 20 plus 20 experiments to get to that point. And as you know, as you see from the almighty eye, that the high peak is far away from where you end up. Now, if you do this using DOE instead, you make a small systematic set, very amino acid 1 and 2. You look at the data, and you find that the direction in the space, and you do a subsequent round, and you very quickly you identify the peak and go forward from that. So of course, this is just an example in looking at 2 separate amino acids. Now, try to imagine doing this in not 2 dimensions, but 200 dimensions. Now, of course, the space very quickly blows up. So really what we're trying to do is interrogate biological space using what we call infologs. And infologs are predefined nodes in the whatever biological space you define that are designed to capture as much information as possible with as few data points as possible. So as an example here, looking at comparing a typical randomized library. This is out of some library paper on Nature, where they looked at all these different sequences. And when you actually sequenced your data points in your library, you see that they're typically highly clustered and not really well dispersed in the space, both by looking at the global diversity, but also if you look at a heat map for each position by itself. And so the local diversity is also highly correlated, because it's just a consequence of molecular biology and how you build these things. Whereas when you do this with infologs, now because we define every single data point, we'll basically define it so that you have the maximum space interrogation per data point. And everything is equally far equidistant from each other from a global perspective. And also if you look at the local perspective for every single gene variant, you'll get a maximum distribution of your information. And so once you make these data points, your interrogation of this space will be orders of magnitude more efficient than a sort of a random sampling through a classic library. So once you have these data points, you can then use that to build predictive models. So here's an example where in this particular matrix, what we have here is in this example, just 20 gene variants. Each row is one gene variant of this particular protein we're trying to engineer. And each column is now one specific variable, so one amino acid that's been substituted from something to something else. And you build these matrices, we'll design them, we'll synthesize them, and they're very precisely designed and synthesized. And then we test them in the real commercial endpoint assay that the partner or customer cares about in this particular case. You end up with a functional output data, so Y data, and so gene variant number 1 with this particular amino acid substitutions has this particular activity, and so on and so forth. Once you have that, you can just toss that into the computer, and you say, dear computer, can you tell me which variables correspond to improved versus decreased function? Computer spits back a table of regression weights, and the assumption, and then we can use those regression weights to analyze how to further improve functionality. Now, of course, you really require an interaction term between every single node to be simple additive to get a perfect regression model be generated from this. And as we all know, biology is not perfectly additive, but I think it's a reasonable expectation that the additivity norm is somewhere between. It's certainly somewhere where it's matchable. So there's certain interaction terms, and I'll talk more about that in a little bit. Okay, so here's a case study. It's something we did with Pfizer some time ago, and really it's a biocatalysis enzyme. We're trying to optimize this particular enzyme for a specific substrate and for making a pharmaceutical intermediate. So in this particular case, the starting point that Pfizer had was active against several different substrates, and it was unfortunately, the desired substrate was barely detectable activity. And there was really no crystal structure within reasonable distance away. And of course, they wanted to have this thing very significantly increased in kcat over km. Fold here is of course a poor choice of measurements, because if you divide anything by 0, you get a very high fold increase in activity. But we need a significant increase in kcat over km, increase in stereoselectivity for this particular substrate, and certainly increase in yield for this enzyme. So what we typically do, and what we did in this case, we go to a GenBank, we identify all possible homologs in GenBank, and then we align these, and we look for amino acid substitutions that exist within this alignment. And then we score every single amino acid substitution against the conservation, diversity, substitution rates, etc. And then we build phylogenetic trees and identify what all the amino acid substitutions, their presence and their abundance, distributed across the phylogenetic tree. And if there's structure information available, we can use that to identify the proximity to active site, etc. And in this particular case, we ended up with an alignment where we identified around 2,000 amino acid substitutions that were ranked and scored as described above. And this is the first step to decrease the possible space from in the order of 20 to 100 as a starting point to significantly less by solely choosing substitutions that exist within the GenBank defined space. So we end up with something that typically looks like this. On your left-hand side, you get the adaptation score. So basically, how likely this thing is to be able to massage the protein, versus something down here on this axis is something that's typically are sequencing errors or something really odd, prolines in the middle of an alpha helix or things like that. And we just, very agnostically, we just take the top 60 substitutions, and we build a matrix, as I described before. And in this particular case, we made a third of them 3 amino acids away, and third 4, and third 5 amino acids away from wild type. And then we're using DOE with the sign, so make sure that each substitution is equally represented throughout the data set. And of course, this is all in a clean background. And if you take all these sequences now and look at them from a phylogenetic perspective, which is not really a fair assumption because they're synthetically made infologs, you'll see you get a very nice and even distribution across your space. And then we'll take this data and synthesize them and measure them in activity, and what you see is you get if you now do a modeling as described before, looking at predicted versus measured activity, you get some sort of diagonal, and I've circled here the starting point. So basically everything is worse or equal or worse than the starting point, but that's just fine. We can now take this information and we can look at the relative contribution of each amino acid substitution, and you see some of them are going to be really nice. This substitution number 5, for instance, has a positive weight and is fairly defined, so we'll keep that for subsequent round. And these guys down here are negative. We toss those. We'll never use them again. And to cut a very long story, much shorter, if you now line up all the sequences throughout this experiment, we end up with round 1, 2, 3, 4. And what you're looking at on the x-axis is the total number of sequences analyzed, so there's 307, I think. And then the corresponding activity here measured as kcat over km across the rounds. And anyone who looked at protein engineering slides before has seen this slides like this, but I think the one to really point out the difference here is that on the x-axis you're only looking at the total number of 307 samples tested throughout this entire endeavor, which is typically order of magnitudes less than what you typically see on these kind of slides. And the best slide now is fulfilling all the criteria for commercial application. So if you can now take the data, you can model that same thing corresponding data. You see very high correlation with a cross val of 0.7. So in summary, we in this particular experiment, we made 300 synthetic genes and mapped out the sequence space for this particular enzyme and fulfilled all the commercial criteria for this particular biocatalytic process. In another example, we're going to look at the GST enzyme. So GST is an enzyme that detoxifies a lot of synabiotic compounds and, among other things, plants. So this is something we did with Professor Bengt Manevik in Uppsala University. And the starting point here, we looked at the wheat GST from the tau branch of wheat, of GSTs, and identify that sequence and use that to identify homologs and align them just as described before. So we end up making these, in this case, 96 infologs, and they parse out equidistant from each other as described, and synthesize these, put them behind a his tag, and purify the protein, and then test them against the library of synabiotic compounds, in this particular case, a set of herbicides. And then, so basically what we do is we have a one starting point GST from that is an actual gene, and we basically make a small cloud around that of the infologs, and then test those infologs against activity, against the different herbicide substrates. So this is just a list of all the herbicide substrates. So this is regular, real commercial compounds. So what we see is that the output here, that dozen or so compounds that where we found activity. So the top guys here, we had activity in the wild type GST gene. We saw improvement across the genes within the cloud at certain different improvement folds. And the lower part here are new activities we identified within this cloud. So just described before, for the sake of talking about the protein GPS, we now can take these sequences, we can build models. So here's the model for metallochlore, and you see a good, nice diagonal, and you get a cross value here of 0.5. And again, you can quantitate the different distribution weight for the different amino acid substitutions. So same thing for the allochlore. And the nice thing, though, looking at this from a navigating sequence-based perspective, is you can really look at the relative contribution in weight for every single amino acid substitution and see that you have certain amino acid substitutions that, for instance, this one I56-V is positive for both compounds, whereas you had other ones, for instance, this guy P16-A, that is a differentiator. That particular amino acid substitution works really well against allochlore, but it's very negative against metallochlore. Sorry, wrong way. So looking at this from a principal component analysis perspective, what you're really looking at is here now that the different substitutions distributed as a biplot, and you're looking at the diagonal going down this way towards this compound, and the direction here for allochlore. And so C and B is just a reference compound used for GST. And what you can quickly use now is identify certain substitutions that are very good for allochlore but negative for metallochlore, or compounds, amino acid substitutions that are good for both or bad for both, and use these to navigate the space. And you can look at this from a structure if you want to. But I don't really. The point here is that structure doesn't really help you much in trying to understand what's going on, but you really need the heuristic data to build the understanding of the navigation space. So in summary for the GSTs, we used in this case an order of 60 amino acid substitutions, and used that against 14 herbicides to build basically a activity profile towards all these this interaction between the enzyme and compound. And we can use that now to navigate the space for improving for a whole portfolio of different herbicides starting from the same underlying map. And use this now as a really high resolution sequence activity model off of the enzyme and how these different amino acids contribute to activity. So summary slide, the whole point of what we're trying to do is trying to get as much bang as possible for the buck. We want to get as much data as possible from as few samples as possible. So it really is a question of how do you interrogate a multidimensional space with as few data points as possible. So we've been doing this the underlying math is certainly very reliable and it's been around for a century or so now. And it's really nice to be able to apply that into biotechnology using gene synthesis. And it's fast and it's very cost efficient, and it really helps out understanding and map out the sequence activity relationship, both for future protein engineering endeavors and also I think for making some really nice and strong IP claims around your space. So that was a sort of a whirlwind of I think 500 slides in 25 minutes, but I hope you got something out of it. Otherwise you can look at it on YouTube I think starting tomorrow.
So at this point we'll say we're open for questions, and if you have any questions for Dr. Gustafsson, you can go ahead and type them into the question profile in the webinar, and we'll see if we can answer a few of them.
So the question on if this is a worthwhile endeavor for antibodies, I think the key point here for making this, it really goes back to one of the first slides on what you're trying to do and what your assay capacity is. So if you have an assay, if you're looking for binding, for instance, you have a FACS sorter, which is your assay, and then you can easily do 10 to the 9 or even more, depending on your setup. And for approaches like that, I would certainly use one or a couple of different types of antibody libraries. Now, if your assay is looking at the immunogenicity of that particular compound in a biological model, that's very different, because then your assay capacity is in the maybe 10 to at most 100, depending on your system. And I think from that perspective, it's really worthwhile looking at the ProteinGPS to identify the amino acid substitutions in your antibody that may or may not be involved in creating antigenic activity.
Okay, we have our next question is, how many proteins have you been able to optimize with this method, and what's your success rate?
So we've done, I'd say, 1.5 dozen, maybe 2 dozen different proteins at this point. Of course, most of them are done with corporations, and they tend not to be too happy about publishing this. So we're very fortunate to be working with Pfizer on the one example I did, and the other one was with Bengt Mannewick, who's an academic. Success rate is that's a good question. It kind of depends on how you define success, of course. But in one example, so I would of course say that we have 100% success rate. But of course, in any collaboration, it depends not only on the engineering technology per se, it certainly depends on the assay. We have a couple of examples where we've done this, and it turns out that the assay wasn't really measuring what the customer thought it was measuring. And we could optimize for things that we could measure, but it was really hard to optimize for things that we couldn't measure. And another example, we had one project where the customer switched assay midstream because they had some ideas about how to do it differently. Of course, then we had to go back and redo that. So you could argue that the first half of that engineering project was a failure, and the second half was successful. But it's the technology per se is 100% success rate. In the context of a business project, of course, there are many other factors that are involved to make that a success.
Great. The next question is several parts, but we'll get to them one at a time. The first part is, aren't many positions with variability just surface positions that won't have effect?
So let's look at that question specifically. So that's really what we see if you go back to, let's look at one of those models. So what you're looking at here, for instance, is I think the other one. This is nice. So this is the enzyme activity from GST versus this particular substrate. So what you see is that this particular substitution, K42R, for instance, is very negative in all possible contexts. But most of these guys are, they're just neutral. I mean, basically they do nothing, either pro or con. So what we, and then you have these guys that are good. So really we're approaching this from a completely heuristic, agnostic standpoint, where we say we identify our amino substitutions, we test them, and if they're good, we keep them for the next round. If they're bad, we toss them out. If we don't have enough information, we'll keep them around for subsequent rounds, but probably at a lower frequency, and then just crank the wheel at that point. So you're certainly going to get a lot of stuff that are neutral, and some of them may be surface, and some of them may be internal. And that's really the key point here, is trying to put specific weights for every single variable in your data set. And the reason, the driver for that's going to be that you want to capture as much information as possible from as few data points as possible. So what we're looking at here is just 96 genes, but because you're testing every single amino acid in a systematic fashion, not in a non-random fashion, but it's a systematic fashion, that means that you'll get a quantitative measure for every single one of them in as broad context as you can get within that particular data set.
The next two questions are related, so I'll tell you both of them, and then you can answer as you want. The first is, how do you choose which of the 19 possible mutations to make, since obviously you haven't made every mutation at every position? And then a question that's come up a couple times is basically just asking again, how do you select which positions for diversification?
So I kind of jumped through that one pretty fast because there's only so much time. But let's go back to the Pfizer example. So what we're looking at here, this is so in any project where you're trying to identify the variables and find a perfect combination of those variables for whatever you care about, if it's FedEx or if you're working for Morgan Stanley or you're trying to make genes with 2.0. So you always break it down into two pieces. One piece is the variable selection. So how do you know which amino acid or which variables to identify for navigating, in this particular case, amino acids. And the subsequent one is, and then once you have those variables, how do you find the ideal or the optimal combination of those variables? So what you're asking about is the first half of that. So how do you pick your variables that you care about? So what we do here is in this particular example, and this is fairly generic to how we do it, we pick the gene of interest. In this particular case, Pfizer had already a starting point that had weak but measurable activity. So we take that gene, we go into GenBank, we'll download all the homologs. And this is some time ago we did this, so this is probably in the hundreds of homologs. We do the same thing today, there'll probably be in the thousands of homologs. And then we identify, within that alignment, we look at every single amino acid substitution. We say, so position number 1 is going to be a methionine. But position number 2, maybe it was a valine in 80% of the alignment, and maybe isoleucine and methionine in the remaining 20%. So for position number 2, that means we have 3 options. We have valine, isoleucine, and methionine, and so on and so forth, walk our way through an entire data set. So that means we identify all possible amino acid substitutions, and that could be an amino acid substitution, or it could be a void of amino acid substitution, for instance, if you have a deletion at that particular position, or it could be an insert as well. So, but it is a variable that has a local position within that set. Now you have that entire alignment, and then you can start scoring those amino acid substitutions based on various premises. So what we tend to do, we look at conservation, for instance. If you have a position that you have, say, like 80% has a cysteine, and then you have 20% that have other stuff, you probably want to keep that cysteine and stay away from the other stuff. So we give the cysteine a high score, and the other stuff a lower score. And so that's for conservation. We look at information content, we look at diversity, we look at substitution rates, and at this point we have in the order of a dozen or so features, what we call them, that we look for every single amino acid substitution, and we give them a score, then we just mean center and normalize that score, and then average that over all the different features. So, and then we combine that and get what we call adaptation score, which is then somewhere between 1 and 0 for every single substitution present in that entire matrix. And then we're very agnostically, we just say we plot them out, as I show in this slide, and we say, let's pick the top 60. And we really have no preconceived notion that those top 60 here are going to be better than the next top 60, but they're certainly going to be better than these guys, because these guys are typically really odd things. There could be prolins and alpha helix, as I mentioned. There could be sequencing errors. There could be just other families that sort of snuck into the alignment. And but what we do is we identify these 60 substitutions. Then we go back and we build a systematically varied data set and then test them. So the assumption here is that, and this is an assumption, the assumption here is that biology has already seen all possible amino acid substitutions at all possible positions, and the stuff that kills the enzyme is gone. That's gone from the gene pool, and only the stuff that retains adaptability and functionality of the enzyme has been retained, and that's what we score for, and that's how we then combine those into systematically varied data sets.
Great. So another question is, on creating new functions, they wonder if do you ever have to mutate a conserved position?
So we've certainly seen that. So yeah, so we have in the past. So there are two questions really wrapped into one. So how do you find a new function? It's somewhat outside of the scope of this discussion. So if you have, because here we assume we have a function, so we have something we can measure, and by doing engineering now we can make it better, faster, more efficient, different pH optima, temperature optima, whatever it is. But we have a function that we can measure. Now, if you have something that your starting point is something that is below the level of detection, it's sort of somewhat of a very different question, because you don't know if your starting point is just below level of detection, or maybe 200 orders of magnitude below level of detection. And so you have no idea really what you're grasping for. So what we've done in the past, typically for projects like that, is we go into GenBank, being a gene synthesis company, and having access to these code memorization algorithms. We'll just take, say, a plate's worth or a couple of plates worth, so 96 or multiples of 96 gene variants off of GenBank that, for various reasons, fit a family that could potentially do this function, and then screen for that function. And fairly often, but certainly not all the time, we do find activity that fulfills sufficient activity against what we care about to use that as a starting point.
Great. The next question is, how do you calculate the substitution rate?
So the substitution rates for on the nucleotide level. There are lots of, it's done by software, so I don't know exactly the details, but there are lots of people. I mean, certainly the field of looking at the ancestral sequences spend a lot of time and effort. I mean, Eric Gauscher and Banner Lab and those guys have done some fantastic job in quantifying the correlation between amino acid substitutions and nucleotide substitutions. So you can use that. You can use the difference between the nucleotide substitutions from conserved versus non-conserved proteins to identify which amino acids have selective pressures against them and which ones do not. And so I don't know all the details, but we typically just use those algorithms that have already been developed by leaders in those fields.
Great. Well, that was the last question. I'd like to thank everyone again for joining us on our webinar, and thank Dr. Gustafsson for providing a wonderful topic. Again, just to remind everyone, the webinar will be available online, and in the chat box you should have a link for where to find that. Thanks so much for coming. Bye-bye.